Hello students. So we'll start in 5 minutes. Let the student join and we'll start. So I have dropped the links of Google Drive and YouTube links of last lectures. So if anybody has some doubt or any queries, they can relate to that particular video. And all the presentations are uploaded in that Google Drive link. You can access all the presentation from there. So only 10 students joined till now. Just wait for few wait. Let's wait for 5 more minutes, then we'll start. Let them join. Monisha, you have raised your hand now. Do you have any questions? Monisha,
Yes, sir.
you have raised your hand.
No, no, sir.
Okay, okay. So let's start then. काफी कम बच्चों ने ज्वाइन किया यार. So if you guys any doubt from the last presentation to last session, you can ask. Then we'll start the session. If somebody has any doubt, they can ask. Or else then we can continue to the presentation. If you don't have any. Okay, then let me share my screen then. So can I even confirm if my screen is visible or not? So let's start then. Is my screen is visible to all?
Yes, yes, it is.
Okay, then let's start then. So in today's session, we'll discuss about nanopharmacology, drug targeting, and nanotoxicology. So myself, Ankit Malik from IIT and NPTEL PMRF fellow. So today I'm having the last session. I think this is the last session, na? So on 25th you have your exam, 25th October, right? So nanopharmacology. Nanopharmacology is the use of nanotechnology for the discovery of new pharmacological molecular entities, the selection of pharmaceutical for specific individuals to maximize effectiveness and minimize side effects, and delivery of pharmaceuticals to targeted locations or tissues within the body. It is a branch of pharmacology which is gradually emerging with the applications of nanoscience and nanotechnology in the field of nanomedicine. So, drug design and drug delivery to selected targets to improve pharmacodynamics and pharmacokinetic profiles towards safer and effective treatment is known as nanopharmacology. So, nanotechnology and pharmacology have broad area in that it covers drug acquisition, drug storage and dispensing, safety and caregiver exposure during drug administration, need for new protective equipments, inability to retrieve defective drug or chip or robot once administered, defective nanodrug or applications, excess of drug molecules, effects over a lifetime and toxicity, and decay over a period of time, its predictability, unknown interactions, and human environment animal transmissions. Then there are two terms in pharmacology. One is pharmacokinetics and one is pharmacodynamics. So pharmacokinetics is what the body does to the drug, whereas in pharmacodynamics is what the drug does to the body. In pharmacokinetics, there is the four terms ADME. ADME means absorption, distribution, metabolism, and excretion. This is the process what the body does to the drug. And in pharmacodynamics, this is the drug does to the body. Then there are some nanomedicines that we have already discussed, like lipid-based nanomaterials, liposome, stills liposome, solid lipid nanoparticles, lipopoliplex, lipoplex, and in polymeric nanomaterials like polymeric micelle, polymeric nanocapsules, protein nanoparticles, polymeric hybrid nanoparticle, and polymeric dendrimer. And in CNT, carbon nanotubes, graphene and its derivatives, nanodiamond, fullerenes, and gold crystals, upconversion nanoparticle, quantum dots, iron oxide, silicon nanoparticle, and inorganic nanomaterials. This portion we have already discussed in the last session. Then the field of nanopharmacology can be classified into distinct categories like defining targets, development of drugs and carrier systems, studying target drug interactions, monitoring the target drug interactions outcomes. Then this is the defining targets. So there are some unbound drugs and bound drugs and binding sites. So here the drug should go and exactly match the structure of receptor. As you can see, there are some binding sites and the drug. The drug have some intermolecular bonds, binding groups, and binding regions. So otherwise, the drug cannot bind with the binding site of the receptor. It will go and degrade itself if it is not go to the binding site. The efficiency will be less if the target is not matching with the drug. Then this is active targeting, then covalent drug conjugation, passive targeting, and non-covalent drug conjugations are there. In drug and binding site conjugations, these things are there. With various nanoparticles like liposomes, polymeric nanoparticles, polymeric micelle, carbon nanotubes, and many more. Then, then development of drugs and carrier systems. So here we have HSA, human serum albumin. So we can make biocompatible and biodegradable nanocarriers which can be delivered the drug to the targeted binding sites. Here, as you can see, we have antibody binding complexes, drug HSA conjugates, then then this drug HSA complex, drug HSA nanoparticles. For example, we can use human serum albumin as one of the highly biocompatible and biodegradable material as a nanocarrier. So here we can load them with any kind of drug, like any kind of drug, like easily hydrophobic, hydrophilic. So also we can target these HSA to a particular location. Example, if we want to target the nanoparticle to the cancer cells, we can add the cancer specific antibody into that nanocarrier and then target that particular nanocarrier to the cancer cells so that it can go and bind only to the cancer cell and release anti-cancer to the cancer cells, anti-cancer drug to the cancer cells. So moving forward, so let's discuss in brief target drug interactions are pharmacokinetic interactions and pharmacodynamic interactions. As we have already discussed that in pharmacokinetics there are ADME terms are there: absorption, distribution, metabolism, excretion. And pharmacodynamics interactions are receptor interactions, drug interaction, then drug receptor sensitivity, drug transportation. Then usually the binding sites of macromolecules are more hydrophobic in nature than the surface, and so this enhances the effect of an ionic interaction. As you can see here, the target is attached with the positively charged moiety. Then the drug is attached with the negatively charged moiety. So both can interact each other and release the drug as much as possible. Likewise, same thing, positive drug, the drug is attached with the positive moiety and the target is attached with the negative moiety. So they'll go and bind with each other and release the drug as much as possible. So the drop of in ionic bonding strength with the separation is less than in the other intermolecular interactions. So if an ionic interaction is possible, it is likely to be the most important initial interaction as the drug enters into the binding sites. Then there is one thing, quantitative system pharmacology. Yes, spelling of pharmacology is wrong. Oh, sorry for that. So there are two things, vertical integration and horizontal integrations. So as you can see, the interaction of drug with purified components, then cells, organs, animals, patients, then populations. Likewise, in horizontal integration, there are targets, then targets in cellular network, multicellular network. So first we'll go through the chemistry of any drug, then chemical biology of the drug, then system biology of the drug. Then we'll study the pharmacokinetics and pharmacodynamics of that drug, then pharmacogenomics. Then these goals for quantitative system pharmacology will be achieved as systematic, holistic, and understanding of the drug actions. Then monitoring the drug target drug interactions outcomes. Loss of therapeutic effect, toxicity, unexpected increase in the pharmacological activity, beneficial effects, and chemical or physical interaction like IV incompatibility in fluids or syringes mixture. Then nanopharmacology target. We slow release nanopharmacology, controlled release nanopharmacology, and biobarrier penetration nanopharmacology. We'll discuss this in brief now. So, in slow release pharmacology, the slow release nanopharmacology studies the questions on how to realize this slow release and the influences of slow release on the drug metabolism and the therapeutic effects. So, as you can see in this diagram, there are two things, non stabilized and stabilized. Non-stabilized is the rapid release of all the drug or all the encapsulated drug inside the nanocarrier. And stabilized, there is a slow release of the drug. This green dots, as you can see, consider this as a nanoparticle. See, as you can see, it is releasing rapidly by reaching to the liver. And stabilized is a slow release of the nanoparticle by reaching to the liver. So this is a slow release of nanopharmacology. Whereas in controlled release nanopharmacology studies how to realize the smart release of the drug according to the therapeutic needs in cellular and tissue microenvironments. So it consists of cell sorting, direct migration. They are the mechanism. So morphogen gradients, induced aggregation, adhesive scaffolds, controlled delivery of the drug molecules. Then biobarrier penetration of nanopharmacology. In biobarrier studies the compatibilities of nanodrugs to passing through biobarriers. Then blood-brain barriers, air-blood barriers to realize the treatment of some focal diseases with the traditional drugs can't arrive because of their incompatibility to penetrate biobarriers. Then nanotoxicology. So nanotoxicology is a branch of bio-nanoscience which deals with the study and application of toxicity of nanomaterials. Nanotoxicological studies are intended to determine at what extent their properties may pose a threat to the environment and to the human beings. The size of a particle decreases, its surface area increases, and also allows the greater properties, its atoms or molecules, to be displayed on the surface rather than the interior of the material. The changes in the physicochemical and structural properties of engineered nanomaterials with a decrease in size would be responsible for the number of materials interactions that could be lead to a toxicological effects. So in nanotoxicology, the factors are solubility, surface area, morphology, chemical composition, its size, and its surface charge. So let's start with the surface charge. Here, the aggregation of nanoparticles with biological materials may depend on surface charge. Then solubility. It is if poorly soluble nanomaterials have been shown to cause cancer and can exhibit more pronounced toxicity of that nanomaterial. Then surface area. The greater potential hazard may relate to the surface area of nanoparticles compared with that for the same mass concentration or large particles. Then morphology of any nanoparticle or any nano material. So health effects of nanoparticles are likely to depend their morphology. Then the toxicity of nanoparticles depend on their chemical composition but also on the composition of any chemicals absorbed onto their surfaces. Then the last, size. Size is the key factor in determining its potential toxicity. The smaller particle, the greater it is surface area to volume ratio, and the higher its chemical reactivity and biological activity will be there. Then the toxicity of nanomaterials were broadly classified into two things. One is biological toxicity and environmental toxicity. So let's discuss about biological toxicity. Nanostructures can enter the body via six principal routes like IV, dermal, subcutaneous, inhalation, intraperitoneal, and oral. The entered nanostructures can distribute to various organs in the body and may remain the same structurally be modified or metabolized. So they enter the cells of the organ and residue in the cells for an unknown amount of time before leaving to the move to other organs to get excreted. Then the toxic effects are allergy, fibrosis, deposition in different organs, inflammation, cytotoxicity, tissue damage, RVS generation, DNA damage. Then environmental toxicity. During the outburst of nanomaterial research, there forms a lot of nanoparticles to the environment. Nanoparticle pollution by deposition of nanoparticle in groundwater and soil. Processes that control transport and removal of nanoparticles in water and wastewater are yet to be investigated. Studies on the effect of nanoparticles on plants and microbes are also rare. Then nanomaterial exposure. Occupational exposure and consumer exposure are most highly exposure of nanotoxicology are there. So in occupational exposure, person involved in nanomaterial manufacturing and research with the increasing demand of nanomaterials and market, the exposure of the workers making these materials and using nanoparticles in the manufacturing plants is also increasing. And in consumer exposure, the engineered nanoparticles are used in personal care products, ranging from cosmetics to sunscreens, where decreasing the size active ingredient yields and better performance. Then environmental exposure. In environmental exposure, the engineered nanomaterial applications develop as a projected the increasing concentrations of nanomaterials in groundwater and soil may present at the most significant exposure evidence for accessing environmental risk. Then reason for toxicity. Surface area to volume ratio of the particles which increases their interaction with the surrounding molecules. Chemical composition of the particle which is responsible for its reactivity. Surface charge of the particle is responsible for the electrostatic interactions between the particle and the drug. The complementary of the nanostructures could cause inhibition of enzyme activity either competitive or it's non-competitive. So this much for this topic. So if anyone has any doubt in the entire thing, also from the last presentations, last sessions, last doubt sessions, they can ask. They can drop their question in Dropbox, then ask, then I'll discuss one by one. So those who came late, I dropped the links for the Google Drive and YouTube links. They can access the previous presentation or recorded session from here. I'll drop once again.
Sir, can you talk about
Simon, can you please mute your mic
nanotoxicology?
and drop your question in the chat box?
Okay, okay.
Meanwhile, if anybody has any doubt, they can also ask. Also from nanoparticle things, characterization and synthesis. So you have exam on which date? 25th October, na? If I'm not wrong. 25th October. Can anyone confirm? Nanotoxicity.
28th of October, I guess.
25th of October, na? Okay.
28th.
28th of October. Okay. Cool. So nanotoxicity. Okay, Simon. Let me reshare the slides. See, toxicity is what? Toxicity is किसी चीज का ओवर ले लेते हैं ड्रग टॉक्सिसिटी वह उसी द सेम वे जस्ट इसमें स्क्रीन इज विजिबल टू ऑल ना
It's visible, it's visible.
okay. So if the morphology or surface area, chemical composition is not that much upgraded to the targeted things or any targeted cells, so it may cause the toxicity of any nanoparticle of any drug or of any API, active pharmaceutical ingredient. It can lead to the toxicity. So there are some factors like biological toxicity and environmental toxicities are there. In biological toxicities, like we administer the drug via IV, dermal, subcutaneous inhalation, or intraperitoneal or oral. So some of the nanoparticles can lead to the toxicity, like they have some allergical fibrosis, deposition, and different organs. If in case we have hmm. So if in case the deposition in different organ, if मान लो हमने ड्रग लिया कैंसर सेल्स को टारगेट करने के लिए, इफ इन केस और वो जाके डिपोजिट हो गया किसी दूसरे ऑर्गन पे, अब वो टारगेटेड नहीं रहा, तो वो वेस्ट है वहाँ पे, टॉक्सिसिटी कॉज करेगा, एलर्जी कॉज करेगा, इन्फ्लेमेशन कॉज करेगा, साइटोटॉक्सिसिटी, इट मे कॉज साइटोटॉक्सिसिटी ऑल्सो. So if in some cases it can lead to DNA damage also. So this factors can lead to the DNA nanotoxicity. और कुछ केसेस में क्या होता है, एनवायरमेंटल टॉक्सिसिटी होती है, जो रिसर्चर्स जो साइंटिस्ट जो नैनो मटेरियल पे नैनो पार्टिकल्स पे काम कर रहे हैं. द थिंग इज ऑक्यूपेशनल टॉक्सिसिटी इज देयर. और कई बार जैसे यू कैन सी द नैनो पार्टिकल पोल्यूशन बाय डिपोजिशन ऑफ नैनो पार्टिकल्स इन ग्राउंड वाटर एंड सॉइल. If in case some industries are there, they're outsourcing the waste of the nanoparticle or nanomaterial that will be go and deposit in ground or soil. That groundwater and the soil can be digested and used by some person in the local areas that can lead to the cytotoxicity of that particular nanomaterial and nanoparticles. So there are two things: consumer exposure and occupational exposure. So person involved in nanomaterial manufacturing, like as I said, researchers, scientists, with the increasing demand of nanomaterials in market, the exposure of workers making these materials and using nanoparticles in the manufacturing plants is also increasing. On the other hand, consumer exposure is there. Occupational exposure में वो लोग आएंगे those who are working on it, research scientists, researchers. Consumer exposure में वो लोग आएंगे जो consume कर रहे हैं, those who are the consumer of that particular nano material. The engineered nanoparticles are used in personal care products ranging from cosmetics to sunscreens. Nowadays, the nanomaterials is used in cosmetics and sunscreens a lot. So वो एक बहुत बड़ा reason है जहाँ से cytotoxicity cause हो सकती है. This is the major reason for cosmetics and sunscreens, while decreasing the size, active ingredients, yields, and better performance. So these are some factors for the nanotoxicity. Hmm. Anybody else? So last time I got some requests on email that some of the students are not accessing the presentations on that Google Drive. Are you guys able to access the presentation which are uploaded in that Google Drive now? Just confirm once in the chat box. I've got so many requests to access of that particular drive. Okay. Need to share. Okay. Able to join that. I'll re-upload that presentation once again and I'll access for the viewer only, not to the editor. So you can access, you can type once again then. So there are total four presentation including this will be there. So the DNA, nanoparticle synthesis characterization and data processing. The second one is nanomaterials for cancer diagnosis. Third one is DNA, nanotechnology, and the fourth one is this will become.
Excuse me. I see that there's occupational exposure, nanotoxicity. So, the question is, for
Uh-huh.
how long are these people advised to work in while exposed to this
Just drop your question in
nanoparticles?
chat box, Seema. Your voice is not that clear. Just drop your message in Dropbox.
Okay.
The last one I'll update after this presentation. So you can access from there. This one. Neetu Sharma, na? Neetu Sharma, you can just let me know if अगर देखो आप एक बार वहाँ पे डाउनलोड हो रही नहीं होती then I'll re-upload that presentation. I think वो है access to all है वो. Anybody can access the presentation or that link. I don't know how could you not. So how long it is advised to work while exposed to these nanoparticles? See, there are preventive measures are there. So any things, any work could be there with a prevention. So occupational exposure, like researchers work for 15 years, 20 years, 25 years in the same field in research or in within chemical things. Yes, now okay. So how long it is advised? We cannot say how long we can advise to work in a research area, research. Okay. So what would be the effect of nanourea or such fertilizer? Like how it has effect on the biology of the ecosystem? It is it related to our presentation? Joel Sanjeev. Okay. Is it related to our nanobiotechnology things? So I'll answer it now. Worry. Just wanted. Okay. So nanourea. See, people nowadays ये agriculture में बहुत ज़्यादा आ गया nano field भी. We are also synthesizing some nano fertilizers. So nanourea और other nano fertilizers उसको use करेंगे. The same thing is its toxicity. टॉक्सिसिटी इसका LDHD होता है वो ज़्यादा नहीं होना चाहिए इफ इन केस इट एक्सीड्स तो उसकी टॉक्सिसिटी होगी इट विल कुछ टाइम के लिए इट विल वर्क बेटर लाइक इट विल ओके Simon, let me done with this Joel then I'll start with this Simon. So स्टार्टिंग में क्या होता है हम अगर कोई ड्रग लेते हैं उसकी भी एक ट्रायल रहता है लाइक थ्री लाइक आपने सुना होगा एंटीबॉडी का कोर्स होता है एंटीबायोटिक्स का लाइक 3, 5, 7, 8 7, 10 सो हमें वो कोर्स कंप्लीट करना पड़ता है उस पर्टिकुलर टाइम पीरियड में अगर हम उससे पहले छोड़ देते हैं उसका कोर्स तो वो नेक्स्ट टाइम से वो यूज़ नहीं होती वो काम नहीं करेगी इफेक्टिव नहीं होगी या पर्टिकुलर बॉडी पे पर्टिकुलर बैक्टीरिया को लेके वो जो पर्टिकुलर बैक्टीरिया है उसको लेके ड्रग रेजिस्टेंट हो जाएगा सेम केस इसके साथ है नैनो यूरिया नैनो फर्टिलाइजर्स हम यूज़ क्यों करते हैं यूरिया और फर्टिलाइजर्स एग्रीकल्चर में ग्रोथ अच्छी हो वो थोड़े टाइम पे ग्रोथ अच्छी देगा लेकिन सेम अगर हम सेम उस पे उसका ब्रेक करते रहे उसका जो पीरियड है उसका जो प्रॉपर कोर्स है वो एक टाइम पे टॉक्सिक हो जाएगा उस पर्टिकुलर लैंड के लिए पर्टिकुलर किसी फीड के लिए या पर्टिकुलर किसी भी उसके लिए लगा लो यार ग्रोथ के लिए उस केस में नैनो यूरिया और नैनो फर्टिलाइजर्स इट हैज इफेक्ट ऑन द बायोलॉजी ऑफ इकोसिस्टम इकोसिस्टम खराब कर देगा आप लोगों ने सुना होगा कि कुछ खेती ऐसी होती हैं कि वहाँ पे एक बार अगर हमने ग्रो कर लिया कुछ चीज़ों तो नेक्स्ट टाइम वो यूज़ करने लायक नहीं होती वाली लैंड। सेम केस में वही नैनो यूरिया या नैनो फर्टिलाइजर्स के साथ भी वही है। कुछ चीज़ें टॉक्सिक हो जाती हैं, नैनो टॉक्सिक टैक्सोसिटी नैनो टॉक्सिसिटी बताया आपको, एनवायरमेंटल टॉक्सिसिटी बताई थी आपको, राइट। सो एनवायरमेंटल एक्सपोजर में वही चीज़ which I've told you in environmental exposure. Then Simon Samuel Simon, please come again. I didn't get you. Okay. For how long it is advised to work while exposed to these nanoparticles? So we cannot advise someone to work for a particular. We cannot advise someone for for how long they can expose to this particular nano material things or nanotechnology. It depends on preventive measures. If you are working in a proper prevention things, can work over a long period of time. Okay, Simon. Anybody else? Like last time, some students have doubts from that nanoparticle synthesis characterization part. If you have any doubt in characterization part, you can also ask. This is the last session. So after this, there's no session will be there. So you can ask any characterization or nanoparticle synthesis questions also. So if you guys don't have any questions, so let's conclude this meeting then. Okay then, students. Good luck for exam. So you can.
Thank
Okay, Simon. So okay, thank you, students, and good luck. You can leave the meeting.